Welcome to this evening's Broad Discovery Series event. My name is Tom Ulrich. I'm the Associate Director for Science Communications here at the Broad, and I'm thrilled to have you all joining here in the room, and also folks watching online. If you're not familiar with it, the Broad Discovery Series explores the genetic and biological roots of human health and disease, the mechanisms that govern how our cells and our bodies function, new technologies that are changing what's possible in science, and the progress that's being made to translate new discoveries into treatments for both common and rare diseases. For this evening's event, we've partnered with our friends at the BroadIgnite program, which, if you're not familiar with it, champions the early career researchers at Broad who have bold, innovative ideas. Ann Carpenter, who is an institute scientist, senior director of the Broad's imaging platform, a past BroadIgnite awardee, and the current BroadIgnite co-chair, will tell you a little bit more about the program, and she'll also introduce tonight's speakers, Ralda Nehme and Shiwei Wang, who are two of the program's talented awardees. If you have any questions for our speakers tonight, I encourage you to flag down one of our mic runners during the Q&A portion of the evening. And if you're watching on YouTube, I'll be keeping an eye on YouTube chat. You can type your questions in there, and I'll do my best to try to work them into this evening's discussion. If you're a Brody or a scientist with a deep scientific question, I'm going to ask you to please hold your questions until the reception so that members of the community get a chance to have their questions answered. And speaking of the reception, if you're in the room, I invite you all to join us in the Broad Discovery Center right out here in the lobby after the talks are over to join us for some light refreshments. And with that, Ann, over to you.
Thanks, Tom. Hi, everyone, and thank you to the folks online also who are with us today. I want to welcome you and specifically describe a bit about the BroadIgnite program, which funded the two awardees who are going to be speaking tonight about their important work in brain cell communication. BroadIgnite launched about 10 years ago. It's a philanthropic program that supports innovative early career research. So really going for the folks that are early on in their career, haven't had a chance to really make their mark or try their own innovative ideas. We really want to give them the footing to be able to try something a bit risky and a bit new. And that really that investment really pays off. We have funded 76 projects over the past decade with the program, and we've seen many of the awardees who are graduate students, postdocs, early career go on to become leaders in their fields across the globe. And that's really such a delight to see our impact on global science through this program of awards here at the Broad. In addition to the grant funding that goes to each awardee to support the research that they're most excited about, each awardee has a kind of customized program of career support to help them learn how to speak to audiences better and to convey the excitement of the research that they do, as well as mentoring and that kind of thing. So, introduction to myself, I'm Ann Carpenter. I am an awardee in 2018 through the BroadIgnite program, and then became the co-chair together with Roby Bhattacharya and Bronwyn McInnes. And I'm also a donor to the Broad Institute because I believe so strongly in what we're doing here. And as you'll meet many of the scientists, so if you talk one-on-one, we're just absolutely passionate about what we're doing, trying to make the world a better place through scientific research. Today, I'll just describe a little bit about my own project, which is on this theme as well, in psychiatric disease. The project that I was awarded for was a very out there kind of idea. We were working on looking for the underpinnings of schizophrenia and other conditions that lead to psychosis. And we collaborated at McLean Hospital. Many of you are aware of this really premier psychiatric research hospital that we have here in the Boston area. And our collaborators there, Bruce McLean and Donna McPhee, had collected a group of patients that experienced psychosis and a set that did not. And by taking cells from those patients and studying them by microscopy, we were able to identify through my BroadIgnite project that the mitochondria had an unusual localization in the case of patients that were experiencing psychosis. And we're very excited now that the National Institutes of Health has adopted the project and is now doing drug screens to try to find, are there any novel compounds that might do a good job of shifting that localization and trying to normalize the mitochondria. So really exciting project that would not have been funded by any other route. And that's really the point of the BroadIgnite program. So for our speakers tonight, we have, they'll be sharing the current research project progress that they have from their recent awards, and we'll have a Q&A afterwards. So the theme of tonight is just the flow of information between brain cells. When communication is disrupted, all kinds of things can happen, and we end up with different conditions like Alzheimer's, schizophrenia, bipolar, and so on. And what's really exciting is some of the research that these innovative young people have taken on. And I'll begin by announcing Ralda Nehme, who is an institute scientist and directs the stem cell program at the Stanley Center, which is part of the Broad Institute. And she's going to tell us about new ways of studying brain cell communication and the importance of her findings for treating psychiatric disorders. She is an awardee from 2021 and is studying a particular deletion syndrome called 22q11.2. Perhaps you've never heard of it. Most of us have not. And while it is itself a rare disease, it can inform us about brain mechanisms that are in common across many different disorders, and really she's pushing the field forward in understanding brain development in healthy conditions as well as unhealthy conditions. After that, we'll have Shiwei Wang, who is a graduate student very early in his career in Laura Kiessling's lab across the street at MIT. And he will be telling us about his work in the role of sugars in brain disorders. And all kinds of jokes can be made about how sweet that work is. But he will be telling us about the importance of those sugars as a signaling component for brain cell communication. And again, how does that function in normal brain development as well as in neurodegenerative diseases? He just received their award last year, so give him some time. There will be a lot of potential described in his talk, as well as what he's accomplished so far, developing technologies to visualize and study sugars in the brain. So with that, I'm very pleased to invite Ralda up to begin.
Thank you, Anne, so much for this lovely introduction. And thank you, everyone, for being here today. And I'm very pleased to be able to share what my lab is working on in trying to understand complex brain conditions, such as psychiatric conditions or mental illnesses. So mental illness is really a major global health challenge. Conditions such as schizophrenia and bipolar disorder can cause severe symptoms, including mood swings, hallucinations, and also really increased risk for suicide. And these conditions affect millions of individuals, and they actually contribute to homelessness, to unemployment, as well as incarceration. And really like something to take seriously is that current treatments are really outdated. There haven't been new effective therapies that have been developed. In fact, many of the therapies have not changed in decades, and over half of the people that live with these conditions still really struggle with daily functioning. However, there's really hope on the horizon, because large-scale human genetic studies have recently uncovered hundreds of gene variants that they were able to confidently link to these conditions, such as schizophrenia and bipolar disorder. So for the first time, we know what these gene variants are, these like differences in the genes, these mutations. However, we don't know what these gene variants are doing. We don't know, and we would like to understand how these gene variants affect cells and cellular programs, and how they shape brain development and function, and how in some individuals, when we have these changes, then this leads to disease. So really, my lab is interested in understanding and bridging the knowledge gap between what the gene variants are and what they do at the cellular level. And I think it's fair to say that progress in developing effective therapeutics for these psychiatric conditions has stalled in large part because these conditions are highly complex and heterogeneous. I just told you that this is the case at the genetic level. So we have not only one gene, but hundreds of genes that have been linked to a given condition. But this is also true at the clinical level because patients and patient symptoms are really different from one patient to the other. So the constellation of symptoms and presentations in a given individual with schizophrenia, for example, can be quite different from the other patient. So we really thought to more deeply understand what is going on and how the complexity and heterogeneity might manifest at the cellular level, because we think that once we understand what's not working properly at the level of the cell, then we can develop better ways to correct it. And ideally, to understand this, we would need to be able to study and manipulate the living human brain. However, for many obvious reasons, this is not ethical or also possible. We don't really have access to human brains throughout development, throughout a person's life, and we can't really do experiments on living people. However, what we can do is that we can study human brain cells in the lab, and the way to do that is by actually using human stem cells. So what are stem cells? Stem cells are really special cells that have unique properties in that they can indefinitely divide, so they can make more of themselves, but they can also turn into specialized cells. So they can turn into any given cell in the body, what we call differentiate. So they can become a muscle cell, a neuron or a nerve cell, a heart cell. And another really cool aspect about human stem cells is that so we can derive them from any given individual anywhere in the world. So we can take a blood sample or a skin biopsy. And then from these cells, we can grow them. And by adding special factors to them, we can turn them into stem cells in just a couple of months. And then once we have these stem cells, then we can turn the stem cells into any cell type we're interested in. You'll hear me talking a lot today about brain cells, so we can turn these stem cells into brain cell. And again, the cool thing is that these, when we take the blood sample, for example, from person A and then make stem cells and then make brain cells, during all of this process, we preserve the whole genetic makeup of that individual. So all of the genetic information in that person is still captured in the derivatives that we then study. So in order to enable the kind of approaches to be able to study complex psychiatric conditions in cells from many different people with and without the diseases, we needed to first develop several tools and resources. And we are fortunate to have been able to do that here in our lab at the Broad Institute. And I'll use this one slide to take you through kind of three kind of critical resources and tools that we've developed. So the first one is that we assembled through a large international collaboration. We assembled stem cell lines from over 1,000 unique individuals, 1,000 donors. And for each and every stem cell line, not only have we really kind of grown very high quality stem cells and we're able to freeze them and bank them here at the Broad, but we also, for every single stem cell line, we have generated genetic sequencing data so we can link the genetic information to what we observe at the cellular level. The other key thing is that we also, over the last few years, have been able to develop ways to turn these stem cells into different types of brain cells very effectively. So we can now take stem cells from hundreds of people and turn them into specific brain cells. We can do this very well and at scale in the lab. And last but not least, I've just told you that we have now a resource from over 1,000 stem cell lines from 1,000 people. But actually, stem cell work, the kind of work that we do, is very expensive and it's very time-consuming. So if we wanted to examine cells from each of these people carefully, all at once, it could take us a really long time, and it could cost us a lot of money. And it also, each time we start an experiment with one person's cells separately, we could have some what we call technical variability. Each time we start an experiment, there could be little differences that we might not be aware of, and we have to take into account in our analysis. So to get around this and be able to study cells from a lot of people all at once, we developed different tricks to do that. And one I'm highlighting here is called the Cell Village experimental system. In this system, we can mix cells from many different people all together in one dish. So instead of having 100 dishes to study cells from 100 people, we can have just one. And that way, all the cells are exposed to exactly the same conditions in the same dish, and we can then score them for different behaviors. And then we actually can then use the fact that each person, the genome of each person is unique. So it's almost like a barcode that we can then use on the back end to identify the person that the cell belongs to. Okay, so now that we have these tools and resources in hand, how can we then leverage them to unravel the complexity of psychiatric conditions? If we revisit all what human genetics have taught us when it comes to schizophrenia and psychiatric conditions, we can see that there's a very strong signal that emerges, and that is that genes that encode synaptic proteins seem to be highly involved in these conditions. So what are synapses? Synapses are actually the connections between two nerve cells or two neurons. And this is how neurons communicate or talk to each other in the brain. So one neuron, neuron 1, which is also called presynaptic neuron, will send a chemical signal to neuron 2 or postsynaptic neuron. And that's how they talk to each other. And the field has largely focused over the last decades on really studying neurons when it comes to psychiatric conditions and schizophrenia. However, we wondered if other cells could also be involved in this process. And we know, for example, that there's one important cell type that is present at the synapse, and this is the astrocyte cell type. And astrocytes are basically, they make up almost half of the cells in the brain, and they are not neurons, but they provide really important support for the neurons. They enable neurons to develop and mature properly. And again, this is what they look like. They're beautiful cells, and they are a key component of the synapse. So they are really essential for synapse or communication between neurons to happen properly. So we wondered if astrocyte-neuron interactions, the way these two cell types talk to each other, if these could be different in psychiatric conditions. And so we wanted to study that. And how do we study astrocyte-neuron interaction in the dish and the lab? And so we can we can grow neurons and astrocytes in the lab. We can grow them separately, each cell type individually, in what we call monoculture, or we can grow them together in the same plate so that they can touch each other. And this is called a co-culture format. And so we did this. We studied cells from roughly 80 people and their neurons in the dish. And then what we found was that when the two cell types touch each other, when they make physical contact, we observed that at that time, synaptic genes are increased in their expression in neurons. And that was perhaps not too surprising based on what we knew about synaptic biology. But the surprising part was that actually when the two cell types touch each other, other genes are really dramatically increased in astrocytes as well. So there's a lot of changes in the astrocyte cell type. And these genes included genes in cell adhesion, so genes that enable cells to stick to each other, as well as genes in cholesterol production. And that was surprising. Now we see that when the two cell types touch each other, cholesterol is increased in astrocytes, as well as sticky genes, basically. And I want to highlight two such genes here. The first is orexin-1. And orexin-1 is known to be a high confidence, so we're quite sure that it is involved in schizophrenia, but also in autism. And it has been studied for many years in neurons, but here we're showing that actually this gene is changed in the astrocyte cell type. So again, further evidence that merits to be studied in this cell type. And then the second gene that's changed is HMGCR, which this is a key enzyme in producing cholesterol in astrocytes. And coincidentally, soon after we had made these observations, similar observations were being made from studying the human brain, but by our colleague, Steve McCarroll, here at the Broad. And in a study led by Emmy Ling in Steve's lab, they studied roughly brains from 200 people after they had died. And they found that these exact gene expression programs were also coordinated or co-regulated across neurons and astrocytes in the human brain. So not only in the dish, but also in the human brain. In postmortem brains, you can see the same interactions. They actually termed this program the synaptic neuron astrocyte program. And then they went on to show that these expression programs were reduced in the brains of schizophrenia individuals. All right, so what is astrocyte doing there? So it turns out that astrocytes are the major source of cholesterol in the brain. Astrocytes make cholesterol and then secrete it outside the cells, and then it's taken up by neurons, and it becomes a key component of the membranes in neurons, and thus it contributes to synapses. Now, when we looked at the genes that are involved in making cholesterol in astrocytes and pumping it out of the cells, or at the genes that are involved in taking up cholesterol by neurons and storing it, we noticed that for many of these genes, they happen to be near regions of the genome that are associated with schizophrenia. So then we wondered, well, what would happen if then we prevent neurons from taking cholesterol that's made by astrocytes? So we can do that by using a gene editing technique called CRISPR interference or CRISPR-I. So by doing this, then we block the expression of the receptors that take up cholesterols. And when we did this, we observed that there's now less expression of synaptic genes in neurons. So this further solidifies this link between cholesterol production in astrocytes and synaptic gene expression in neurons. And from there, we now wanted to understand if these genetic variations that have been documented by the genetic studies to be present in individuals with schizophrenia, if these may affect how astrocytes and neurons talk to each other. So to study this, we first started by examining one genetic variant that is considered to be the highest genetic risk factor for schizophrenia. And this is the 22q11.2 deletion that Anne mentioned. So this is actually not only that does it confer a high risk for schizophrenia, it is actually one of the most common, it is the most common human chromosomal deletion in humans. So this removes a part of chromosome 22. And we were able to assemble a cohort of stem cells from individuals that either carry or do not carry the deletion, and for the ones who carry, that also have schizophrenia. And what we found was that when we looked at their gene expressions, we found that in neurons, the genes that have been linked by the genetic studies to schizophrenia and that are also synaptic genes, we saw that they are decreased in their expression in the neurons. And in a more recent study, we also showed that the astrocyte component of the synaptic neuron astrocyte program, that was also decreased in astrocytes that carry the deletion. So now we wanted to also understand if we could detect changes other than just how the expression levels of the genes. So we wondered if we could really measure how these genetic influences, these genetic risk factors change how cells look or cell morphology. And to do that, we took advantage of a wonderful assay that has been developed here by Anne Carpenter and Stu Schreiber here at the Broad, and that Anne mentioned in her talk. And in this assay, cell painting, you can use different cell dyes to label different cell compartments or organelles. And so we adapted this assay to make it specific to brain cells, and we called this neuropainting. And then we screened neuronal progenitor cells, neurons, and astrocytes from our cohort with or without this risk factor, the 22q11.2 deletion. And when we did this, we were able to identify that there are many traits, many morphological features, as we call them, that are different in stem cells, in progenitor cells, in neurons and astrocytes. Some were similar across all the different cell types, and some were unique to each cell type, these differences. And I wanted to highlight one here, that is a change in how the ER, which is one organelle, and the mitochondria, how these two organelles are distributed in the cells. You may be able to appreciate here how their distribution in control cells, so cells from people who do not carry the deletion, versus cells who carry the deletion, how these look different. They are distributed differently in the cells. So the deletion is affecting that. And then we went on to link these changes in how the cells look and how these organelles are distributed, we could link these to changes in an expression of specific cell adhesion genes that make cells stick to each other. And because of all of our findings on cholesterol, we actually then wondered, well, can we look at, cholesterol or lipid features in this system? So we then incorporated lipid profiling. We looked at lipid expression into our neuropainting assay, and then we screened our cohort again. And what we found was that actually, just by looking at lipids alone, by looking how these lipid-related traits are distributed in the cells, we could tell whether a cell has the deletion or not. So I'm sorry here, the graph is a little messed up here, but in this case, it shows that there's much fewer lipid spots in the deletion versus without it. And so the last vignette I'll tell you about tonight is about our work on antipsychotic medications. So we wanted to understand then can drugs that are typically used to treat psychiatric conditions like schizophrenia, can these change how neurons and astrocytes talk to each other, or what programs are involved there? And so to do that, we then took neurons and astrocytes that were derived from many people, 140 people in total, and we treated them with many different antipsychotic medications at different doses. And to make a long story short here, I wanted to highlight one main finding from the study, and that is that the antipsychotic clozapine, which is one of the most effective antipsychotics, and I'll come back to that clozapine increases cholesterol production by astrocytes. And we found that this was true in cells from every single person we looked at. So in this plot that I'm showing you here, each color corresponds to cells from a different person and how much cholesterol is expressed in the cells from that person. And as you can see, the condition in the middle here is clozapine. And you can see that when we treat cells with clozapine, we see much more cholesterol in cells from all the people we looked at. So as I mentioned, clozapine is a really effective antipsychotic, but people don't really understand why it works so well. And so we wondered if effects on cholesterol production may provide an explanation. And so when we, again, examined our gene expression data in our co-culture system, we found that not only does clozapine increase cholesterol production in a nice dose-dependent manner, but it also increases the expression of synaptic genes in neurons also in a dose-dependent manner. So here, by manipulating the levels of cholesterol in clozapine, we can also see a corresponding increase in synaptic gene expression in the neurons. And the kind of last thing I'll show you is that, well, what happens if we add clozapine to then these the 22q11.2 deletion cells? I just told you that we can see that there are many changes that we can measure in how cells look in cells with this deletion. And well, we found that clozapine, at least to some extent, appears to rescue some of these morphological defects that are related to the 22q11.2 deletion. And again, so clozapine is not an ideal drug treatment. It doesn't work for everyone, and it also it's associated with many side effects because many metabolic side effects. And in fact, it turns out that a lot of the patients that take clozapine have increased levels of lipids in their plasma, perhaps related to what we're showing, again, effects on cholesterol production. So can we then use the tools we develop to then develop eventually better treatments? So to sum up, I've told you today that astrocyte-neuron interactions play previously unappreciated roles in psychiatric disorders. The biological programs we discovered are consistent with genetic studies and are altered in cells from people who harbor variation that's related to psychiatric conditions. And finally, the fact that we can manipulate these programs in living human cells and modulate them with drug treatment holds great promise for therapeutic discovery. And I'll end by thanking you for your attention, thanking everybody who has contributed to this work, especially people in my lab. A couple of them are present here. I'll give a special shout out to Derek Hawes, who has been my co-conspirator, our lab manager since I started my lab, and Jack Stu as well, who's here and contributed to this work. Our funders, obviously, BroadIgnite and other funders, and here are the people who did the work. Thank you.
And we'll wait for questions till the end. We'll next give Shiwei the chance to speak.
Okay. Hi, everyone. Thank you for being here. My name is Shiwei. I'm a PhD candidate at MIT. I'm also a BroadIgnite awardee at the Broad Institute. And today it's my pleasure to be here to tell you about our work to unveil brain cells' sugar-coated secrets. So I want to begin my talk in the country of Colombia, where a family suffers from a rare and devastating condition. So around 1,000 people that lives in central Colombia, almost everyone develops Alzheimer's disease. And not only that everyone develops Alzheimer's disease, they develop Alzheimer's much earlier than the general population. And a doctor has been working with this family to provide care and to find out why they're having this condition. And it turns out they have a very rare genetic mutation. And in this family, so this is called a survival curve, where is a way to depict how a population develops disease over time. So when everyone's healthy, the curve's at 100, and as people start to be diagnosed with conditions, the curves start to drop. So as you can see, by their mid-40s, most people would have been diagnosed with mild cognitive impairment, which suggests noticeable cognitive decline and suggests that's an onset of Alzheimer's symptoms. And by their mid-50s, they would have been diagnosed with dementia. So it's a very unfortunate and devastating condition, and a team of Columbia doctors have been working with this family to provide care and to trace the family tree to figure out what is happening. And what's really interesting is that over the past three decades, the doctors start to realize that there's some outliers in this family that develop this disease much later than others. And in particular, there's this woman that's named Illyria. She has this mutation that would guarantee she will get Alzheimer's, but for some reason, she didn't develop any symptoms until 72. So that's almost a 3-decade delay of symptom onset compared to other members of the family. Of course, the doctor and scientists become very interested in this case because understanding what's protecting her could inform us to develop therapeutics that can benefit the over 30 million patients who are suffering from Alzheimer's in the world today. And so unfortunately, she passed away in 2020 from cancer, and she generally donated her brain for science. But while she was still alive, they invited her to come to Boston to get some brain scans in the Massachusetts General Hospital. So this is PET scans of her brain when she was just starting to have cognitive impairment. And the redder brain region suggests she has a higher level of the toxic proteins in that region. So when they looked at amyloid beta, which is the most well-known hallmark for Alzheimer's disease, she has an exceptionally high level of amyloid beta compared to patients from that family in their mid-40s when they're starting to develop symptoms. However, when they looked at the protein named tau, also another well-known marker for Alzheimer's, she has relatively moderate level of tau compared to other patients. And they did more tests, and she showed that she has very minimal cell degeneration, which explains her avoiding the disease. So what this suggests is that for some reason, the disease progression is impeded in her brain, and that causes a resistance to the Alzheimer pathology. And that's really interesting. So the doctor performed all kinds of tests, including sequencing her whole genome to identify, so she has this mutation that causes Alzheimer, maybe she has something else that's protecting her from Alzheimer's. And they found a very interesting hit. So if you're paying attention, so Ralda's talk also mentioned this protein called APOE. So this is a protein that's very important for lipid metabolism. So this protein helps to shift the lipid around your body, including cholesterol. But interestingly, the gene that encodes this protein, the gene APOE, is also the best predictive gene we have for Alzheimer's risk. So when you have a genetic testing based on your gene composition for APOE, they can predict your risk of getting Alzheimer's in the future. So this is a very interesting hit because that suggests this could be a major reason why she has this resistance to Alzheimer's disease. And the mutation in this APOE, which is named Christchurch, happens to be in the region of APOE where the APOE protein uses to bind a self-surface sugar on the brain cells. And indeed, when the researchers tested whether the interaction is intact, it turns out this mutation, which is called APOE3, it's Christchurch, does affect the interaction between APOE and the brain sugar. So this is a biochemistry assay that tests the binding between protein and sugar. However, of course, this is very exciting, which says this is a new direction for us to look into to understand Alzheimer and potential therapeutics. But at the same time, the sugar they use in this case called heparin is only an analog of the actual sugar in the brain, suggests they may not be representative of what's actually happening in the human brain. And also, this is just a biochemical assay, and a lot more work is needed to actually understand the biology of how this sugar works in the human brain. So I want to give a little introduction of the sugar in your brain. So it's not very well known, but all of the sugar in your brain and actually in your body are covered, all the cells are covered by sugars. And I want to introduce a new term here. So for biochemists, when a series of sugars that's connected into an ensemble, we call this a glycan. And every cell has all kinds of glycans on the surface. And they all have different functions, and they all interact with different partners. But very interestingly, APOE mainly interacts with this particular sugar called heparan sulfate. And that will be the topic of my talk. And here's a picture of a group of cells with heparan sulfate visualized on the surface. And you can see we're looking down on this population of cells, and we can see the nucleus where the DNA is, and you can see the heparan sulfate on the surface that's pointing out. And you can see this sugar acts as a barrier between the cell and the outside. And we know that this sugar plays very important roles in the brain. So one example is when your brain is developing. So when your brain is developing from embryo to a person, a lot of communication is required for the cells to know where to go and what type of cell they should become. And here's a study in the mouse brain. If we remove the sugar as a channel for cells to communicate with each other, we actually see major disruptions in the formation of different brain regions. You can see this is the olfactory bulb, and that's completely missing in the mouse that's missing this sugar. And we also observe significant disruption in the connection between different brain regions. So this sugar is critical for brain development. We also know that this sugar is very important for synapse formation. So synapse is how your brain cells connect with each other. And if we remove this sugar, the synapse will become dysfunctional and the signal cannot be transmitted as efficiently. And here we also have the example showing this sugar is very important in the process of neurodegeneration. Understanding that can be very valuable. However, our understanding is still quite limited, mostly because we don't have good tools to study the sugar in the brain. If you notice how I describe these studies, the best tool we have right now is to remove this sugar from its biological context and see how the system dysfunctions. However, just by removing it, so say this is important because the system's broken, really doesn't help us to fully understand what it's doing in the brain. So what my PhD is focused on is to develop tools to actually start to visualize this sugar in the brain context so we can understand how it works. And the technology that we use is called expansion microscopy. So the major challenge of studying molecules in the brain is because they're too close to each other to be distinguished from each other. So why don't we just move them apart from each other? So that's what this technology does. By forming a polymer that's commonly used in baby diapers and contacts in the brain tissue, we're able to physically magnify the brain by bringing all the molecules apart from each other while still preserving the overall organization. So now using a conventional microscope, we can get state-of-the-art images of how molecules are organized in the brain tissue. And we apply this technology to visualize these sugars. So this is human pluripotent stem cell. It's a cell that's in the development stage. And I showed this image before. So you can see this heparan sulfate glycans as very hairy extended structures on the cell surface. And very interestingly, when we differentiate these cells into motor neurons, so neurons that's more in the terminal state, all of these sugars become very curled up on the cell surface. And this is very surprising because for a long time, scientists have always thought these sugars are just floating uncontrollably on the cell surface. But what this means is that the cells actually control the structure of these sugars on the surface to make sure that they function as they're intended to. However, these samples are expanded around fourfold, so it's still quite limited in terms of resolution. So one thing I developed during my PhD is I engineered a polymer that's used to expand these tissues. So this is just a pure polymer. We will, in reality, we'll embed biological samples in these hydrogels to actually blow them up. But you can see this is the same gel before and after expansion in the same dish. So this gel is able to expand up to 20-fold while still holding its shape and holding all the spatial relationship intact. And with the more advanced expansion technology, we're actually able to start to zoom in on the fine details of molecules. So this is different synapses in the mouse brain. And I zoomed in onto 3 different synapses. So we mentioned the synapses connection between neurons. So there's a cell that's sending the signal, and there's a cell that's receiving the signal. And if we look at the proteins that's sending the signal and the proteins that's receiving the signal, we see the shape of the synapse very clearly with this technology. And we can also see the space between the synapse. That's the distance, the signal that needs to travel. So we start to apply this technology to the sugar. And another advancement of the technology we start to use is to recognize that actually there are different flavors of this same sugar. So it's the same type of molecules, but they actually go through extensive modifications, so they interact with different kinds of partners. And we haven't really had the ability to recognize how these different flavors work in the biological context. So we start to apply these different protein probes that can recognize these different flavors in the same cell to start to look at how these different sequences distribute. So on the left, we have 2 different probes on different population of cells, and on the right, we have a different probe. And then just visually, you can see there's a difference where these 2 probes start to recognize these glycans that's pointing outside of the cell. These probes only looks at glycans that's very close to the cell surface. And when we zoom in onto this population, you can see these 2 probes really don't overlap in terms of what they recognize. And when we expand these samples 20-fold, what we start to see is that these different sequences, different flavors, they actually arrange spatially different along this one glycan. So what that suggests is that not only the structure of these glycans are controlled on the cell surface, even these different modifications are very carefully placed on the glycans by the cells, which was previously not observed. And what this really shows is that there's a very complex machinery that the cell deploys to control these sugars to accomplish the critical roles they need in terms of cell-cell communications. And on top of that, we start to apply this technology to study Alzheimer's. So this is the same human brain tissue from a patient after they passed away. And we're starting to visualize this sugar, heparan sulfate, as well as other proteins that's significant for Alzheimer's. And what we're starting to visualize is that this sugar is co-localizing with the key players of Alzheimer's. And we're gonna look into how they're interacting, and also look into what other potential partners like proteins the sugar is interacting with. And if we disrupt the APOE sugar interactions, like in that patient, how will the system change? Which will hopefully lead to a deeper understanding of the disease and well as potential therapeutic directions. So in conclusion of my talk, I would like to present 3 takeaways. The first is every cell in your brain is covered by sugars, and evidence indicate that they play critical roles in cell-cell communications, as well as diseases such as Alzheimer's disease. Our understanding of biology and disease is driven by the development of different tools, such as the tool of physically enlarging a brain to look at things clearer. And breakthrough in our understanding, our sincere hope is that that can lead to diagnostics and therapeutics for different diseases, such as therapeutic, cancer, and even potentially aging. So with that, I would like to thank my PhD advisors for providing a great opportunity to work on these problems. I want to thank our collaborators also both in the lab and around the world. And I want to thank all the donors who donated their body for science, and also all the funding sources, especially BroadIgnite, which enabled this work. Thank you for your attention.
Wonderful. Thank you so much. I find this really fascinating as an example. The pair of you are, and please get ready to ask questions, raise your hand, and a microphone will come to you. I just find it fascinating that there are so many different styles of science. So you are clearly zooming in very, very far to look at the synapses at the tip of a little arm that is at the end of a very large neuron inside a brain, inside a human. Whereas Ralda, you're sort of zooming out. Let's get this population of patients and look at lots of different samples and to look at the structures there. So I just find this really fascinating. The one thing you had in common, though, is each of these projects involved quite a cast of characters, and I think that's pretty typical. So let's turn to questions from the audience.
Very interesting from both of you. I was wondering when you mentioned the cell colonies or villages, how the mixture of cells from different people in the same dish, whether these cells interact in some way, and how do you account for that?
Yeah, so it's something that we are studying, right? We can take a little bit to our advantage if we kind of study how cells from a person that don't have a particular genetic variant, how they interact with the cells with a person that has. So these kinds of interactions we can study if we can control them. But for the most part, in the village system, we study what we call cell intrinsic behavior, so behavior within each cell. So when it comes to the gene expression programs, we tend to avoid looking at what is expressed outside of the cells or secreted or cell extrinsic behaviors, because this is too complicated to measure with the village system. That's a good...
Rocky may affect your result.
Yep.
Was it that?
So the kind of really important thing, which I didn't really discuss, is that we also have repeated a lot of what we established for the system. And whenever we make a finding, we also repeat the same experiments to what we say validate them, to make sure that we make the same observations when we culture cells from each person separately. So yeah, absolutely.
I totally understand your research is extremely necessary in both your cases, schizophrenia, Alzheimer's. My question is this pie in the sky kind of research, or how far are you from practical, real world therapeutics? I mean, is it, are you at the nascent level? Are you 20 years away?
I love this question. So let me first give some context that it currently takes somewhere between $1 and $6 billion to develop a new drug. As you can imagine, the Broad's entire operating budget is not quite that huge. And it typically takes something at an absolute minimum about 10 years, typically more like 15 to go from an idea of what might make a good drug and onward. So the Broad usually sits at pretty early in that spectrum, but I'm very curious to hear the answer of what are the next steps, and kind of how long do you foresee till something could come of your research in patients?
Yeah, maybe I can start by saying that. So in psychiatry, for example, for schizophrenia, in the last 50, 60 years, there really have not been any major new drugs that have come on the market. And this is for many reasons. Like I said, because these are brain diseases, it's very hard to access the human brain. And we know people have typically studied mice, for example, animal models, but there are so many differences in both the genes, but also kind of how the brain, structurally, and mice and humans, so that we really needed a human system to be able to study these disorders. I mean, not exclusively, right? But this is one of the reasons. And the other thing here is that with the tools and technology, for the first time right now, we really have the tools in hand to be able to study such complex disorders. So with assays such as, the cell painting that Anne has developed first and that we are really trying to leverage, we can cheaply and quickly screen. So now we know, we are starting to understand what to look for, what are the defects at the cellular level. So now we actually can screen drug libraries to try to identify ways to correct them, and then hoping to get closer to a treatment that actually works in patients, right? So there's always back and forth, but we hope that with the tools present today, that we are getting closer.
Yep. And I can answer from the perspective also of seeing startups that incubate and launch out of the Broad. When you do a drug screen and get a promising first chemical, it usually takes a couple of years to optimize the chemical. And then it takes a couple of years to test its safety in usually mouse models, but now other alternatives, but still a couple of years. And then you file with the FDA and go to human trials. So it's probably about, let's say, 5 years total from having a good chemical starting point.
Yeah, and I think I would add that if you have been following therapeutics in the Alzheimer's space, I think there have been several drugs that's been approved, but there's no evidence showing that they're super effective. So I think a lot of what we're doing is also like we want the cure as fast as possible, but at the same time, we need to do it the right way and then to make sure we actually fully understand the disease to know what we're going after.
Once it gets to human clinical trials, that can be usually years, especially if it's a disease that takes a while to develop. It takes a while to know if your drug is working. So if it takes several years and usually tens to hundreds of millions of dollars for that human clinical trial stage, we don't want to blow it having messed up at an earlier stage. That's a really great point. Another question? There was one here.
Yeah, thank you both of you for the wonderful presentation. My question is very simple. How do you convert a stem cell into a neuron or any other type of cell? So you can take a stem cell and make it into any other form of cell. How do you do that? What's the process?
Yeah, so actually we can add to the stem cells, we can try to make some genes become expressed and other genes stop to be expressed. So we know what these are based on our understanding of human development, human brain development. And so we can, what we call our transcription factors. So these are like gene products that control how other genes are expressed. So we can add these transcription factors to the cells, for example, and push them to become neurons, or we can block other genes from being expressed by adding some chemicals or small molecules.
Here?
Hi, you mentioned that clozapine, by increasing cholesterol production, affects neuron communication. I was wondering, does it distinguish between low and high density cholesterol and other aspects of, are lipids somehow involved in a communication?
So this is something that we have not really studied, what we call lipid species. I have to say that the kind of version of cholesterol that's present in the brain is a little different, right? But what I can say, I mentioned that clozapine is associated with side effects in patients, and these are often metabolic side effects, including more lipids in their blood that can be measured. So my guess would be that it does not. So these side effects are because they affect the heart. So we aspire and we hope to be able to develop a drug that would kind of increase cholesterol in the brain, but not somewhere else. So that would not cause these, heart issues eventually. Yeah.
Great. And we'll take one last question here. Thank you.
Hi, very interesting talk. Thank you so much. I had a question around clinical trials. So if I understand correctly, determining endpoints is somewhat difficult in such diseases like Alzheimer's. So some of the techniques you are developing, would that make it easier because now you have more of an analytical techniques to see what exactly is happening with the brain cells?
And can you describe what is an endpoint, a clinical endpoint?
I guess I'll start, but then you can tag team. So I think, exactly. This is, so by basically being able to quickly be able to access cells from different people that have or do not have a certain genetic, like a certain mutation. For example, our hope is that we can screen quickly because in the dish, it takes, just only a few weeks to be able to screen. We can measure how the communication between the synapses between the nerve cells, we can count them, we can measure the cholesterol production, and we can then quickly screen for drugs. So these are the endpoints or the phenotypes that we measure, or the sugars, right?
I think just to disclaim, my PhD is not in clinical science. I don't know as much about clinical trials, but I would say, I think just more methods to analyze what's happening in patients, in model systems, would help us to at least have a better idea to set an endpoint before the trial begins. Because I think that's the challenges you see with Alzheimer's, is that a lot of clinical trials were complete first, and then they start to adjust the criteria to make the drug effective. which is not science supposed to be done. Like, I understand clinical trial is complicated and real world changes things, but at the same time, like, that's what we should avoid.
Wonderful. Thank you so much for sharing your science clearly tonight. Please join me in thanking these folks.